Hello, today I'm Nagar, and today we're going to be solving October-November 2022 Paper 1 Variant 1. And before we start, I'd highly appreciate it if you would take a moment to subscribe to this channel and like this video. For free Topic 1 notes, all you have to do is fill in the form in the description box down below. Question number 1. The photomicrograph shows parts of a plant cell. What is the magnification of the cell? So the equation to calculate magnification is IM. Therefore, you divide the image size by the actual size. Right here, they've given us the scale for the nucleus of the plant cell, which is 5 micrometers. And measuring over this on A4 paper is going to give us 2 centimeters. And to calculate the magnification of the entire cell, you don't actually need the size of the entire cell, because as you all know, magnification is a ratio. So it's enough just having the ratio of the nucleus. Now, what we have to do is, before we use this equation, that we convert 2 centimeters into micrometers. How we do this is we multiply 2 by 10 to convert from centimeters to millimeters, then multiply by 1,000 to convert from millimeters to micrometers, which gives us in return 20,000 micrometers. Now, image size divided by actual size, so 20,000 divided by 5 micrometers, which gives us D. Question number 2. Which row matches each organelle with its function? Microtubules. So microtubules are proteins made of tubulin which form the cytoskeleton of the cell and the intracellular transport system. Therefore, let's see the suggestions that we have here. Microtubules make cilia and the spindle. This is correct. Microtubules does make the basement membrane of the cilia, and they make the spindle during mitosis. For B, move vesicles within the cell. We've just mentioned that microtubules form the intracellular transport system. Therefore, they are able to move vesicles within the cell. Now for C, move chromatids apart during anaphase. As we all know, microtubules form the spindle, which indeed holds into the kinetochores of the chromosomes during anaphase and pulls the chromosomes apart. Therefore, this is correct. For D, forms part of the cytoskeleton. We've just mentioned this point, so these are correct. Now, rough endoplasmic reticulum. The function of the RER is, we all know, to synthesize proteins. Now, it separates some processes from the cytoplasm. This is correct. It allows it to form in compartments. Now, assembles amino acids to make proteins. The wording of this is incorrect, because what actually assembles amino acids to make proteins are tRNAs, because we all know that tRNAs hold an amino acid and then binds to a complementary base pair on the mRNA molecule. Therefore, what actually assembles amino acids is the tRNA, not the rough endoplasmic reticulum, so this is incorrect. Now, modifies proteins which may be released from the cell. Now, what modifies proteins is the Golgi body, so this is incorrect. For D, makes triglycerides and phospholipids. This is also incorrect. As we all know, rough endoplasmic reticulum synthesizes proteins, not triglycerides. Now, Golgi body makes lysosomes. Yes, this is correct, because as we've mentioned earlier, it does modify proteins, therefore also make lysosomes, which are enzymes. Now, contains enzymes for destroying worn out parts of the cell. This is incorrect indirectly, because lysosomes is what destroys worn out parts of the cell. So not directly the Golgi body. The Golgi body does synthesize the lysosomes, but doesn't directly do this function. Now, makes glycoproteins. This is correct, because it does modify the proteins. Modifies proteins by adding carbohydrates. This is also correct, which also makes glycoproteins. Now, centrioles. A, assemble the spindle during nuclear division. This is correct, because during mitosis, what happens is that the centrioles are found inside 2 centers called centrosomes. The centrioles are found at right angles inside the centrosomes. These centrioles synthesize the spindle, which indeed attaches to the kinetochores of the chromosomes, separating them apart during anaphase. So this is correct. Now, move chromosomes apart during anaphase. Not directly, it's the spindle, not the centrioles. The centrioles is what synthesizes the spindle, which does this function. Therefore, this is not directly correct. Centrioles move cilia. This is also incorrect. It's the microtubules. Move individual cells. This is also incorrect. Therefore, the answer is going to be A. Question number 3. Where are ATS ribosomes found? So, ATS ribosomes are found in the cytoplasm of eukaryote cells. So, let's see the suggestions that we have here. 1. Chloroplasts. Now, the chloroplasts are actually prokaryotic origins, therefore they have 70S ribosomes. Therefore, this is incorrect. 2. Cytoplasm of eukaryotes. This is correct. 3. Mitochondria. No, they have prokaryotic origins, therefore they have the 70S ribosomes. Therefore, this is also incorrect. 4. Cytoplasm of prokaryotes. Cytoplasm of prokaryotes have 70S ribosomes, and this is also incorrect. Therefore, the answer is C. Question number 4. Which feature, when present in an organism, enables it to be identified as a prokaryote? Now, let's see the suggestions that we have here. A. Cytoplasmic DNA. DNA not bound to a nucleus, not found within a nucleus. This naked DNA in the cytoplasm is a clear indication of a prokaryote. Now, for B, cell wall. Not ideally, because fungi, which are eukaryotes, also have a murine cell wall. Therefore, this is not always correct. C, nucleus. We've just mentioned that in a prokaryotic cells, they do not have a nucleus and they have a naked DNA in their cytoplasm. For D, ribosomes. Yes, prokaryotic cells do have ribosomes. However, also eukaryotic cells have ribosomes. Here they did not specify whether it's 70S or 80S ribosomes. Therefore, this is too vague and cannot be correct. Therefore, the answer is supposed to be A. Question number 5. What is the function of a nucleolus? So, a nucleolus is a structure found inside the nucleus of a cell. Therefore, its function is the synthesis of ribosomal RNA, which indeed combines with proteins to form the ribosomes. So, its function is the ribosomal synthesis. Now, let's see suggestions that we have here. The formation and breakdown of the nuclear envelope. This is incorrect. What's responsible for this are structures, no need to mention their names, on the nuclear double membrane. When these structures are phosphorylated, therefore the nuclear membrane starts breaking down because it becomes soluble when it gets phosphorylated, and therefore the nuclear envelope breaks down. Therefore, this is incorrect. B. The formation of the rough endoplasmic reticulum. No, this is also incorrect. Its function is the synthesis of ribosomal RNA. C. The synthesis of ribosomal proteins. Now, proteins is incorrect. We just mentioned it's ribosomal RNA and then it combines with proteins to form the ribosome. It does not form the ribosomal proteins. Therefore, the only correct answer here is D. Question number 6. The diagram shows the results of number of tests on a solution of biochemicals. Which substances are present in the solution? Now, let's see what each test actually tests for. Now, Benedict's test is used to test for reducing sugars. And a positive result would be from blue to brick red. Therefore, this is a negative result for reducing sugars. Now, non-reducing sugar test is, from its name, used to test for non-reducing sugars. And a positive result would be from blue to brick red or any color along that spectrum. In this case, it's yellow. So this is correct. It does indicate the presence of low concentrations of reducing sugars, but it's still a positive result for non-reducing sugars. Now, iodine test, blue-black. So, an iodine test is used to test for the presence of starch. And a positive result would be from brown, which is the color of iodine, to blue-black. Therefore, it is positive for starch. Biuret test is used to test for proteins. And a positive result would be from blue to purple. Therefore, this is negative for proteins. Therefore, what is positive or present in the solution is going to be non-reducing sugars and starch. Question number 7. Which diagram shows the ring form of beta-glucose? So ideally, you should memorize the formula of alpha and beta-glucose. In beta-glucose, the hydroxyl group on carbon 1 is always above the ring. And in alpha-glucose, the hydroxyl group on carbon 1 is always below the ring. And of course, the hydroxyl group on carbon 3 is always upwards. Therefore, this is the formula of beta-glucose and you should memorize it. Therefore, the answer is going to be A. 8. Which row correctly matches the example with the type of molecule? So disaccharide. Disaccharide is made of two monomers joined together by a glycosidic bond in a condensation reaction. So fructose is a monosaccharide made of one monomer, therefore this is incorrect. Starch is a polysaccharide consisting of many glycosidic bonds formed in a condensation reaction, so this is incorrect. C. So maltose is a disaccharide consisting of two alpha glucose monomers joined together by a glycosidic bond, so this is correct. Sucrose is correct because it consists of fructose and glucose joined together by a glycosidic bond, therefore both of these are disaccharides. Now macromolecule is a giant molecule consisting of many repeating subunits. In this case, glycogen is formed of many alpha glucose subunits joined together by glycosidic bonds, so this is correct. Hemoglobin is made of 4 polypeptide chains with 4 heme groups attached to each one of them, therefore it is technically considered a giant molecule. Ribonucleic acid is consisting of many repeating nucleic acids, therefore this is correct. For D, cellulose is a macromolecule because it's made of many repeating beta-glucose subunits, so this is also correct. Monomers. So glucose is a monomer because it's one subunit. Now ribose is also one unit, so it is a monomer. Sucrose, we just mentioned that it's a disaccharide, so it's not a monomer. And fructose is a monomer, so this is correct. Now, Polymer is made of many repeating subunits. Therefore, starch, as we all know, it's made of alpha glucose, which are many repeating subunits, so this is correct. Glycogen is the same. Cellulose also made of beta glucose subunits. And collagen made of proteins, therefore they are polymers. Therefore, the answer is supposed to be D. Question number 9. The enzyme alpha amylase hydrolyzes amylopectin but is not able to hydrolyze some of its glycosidic bonds. Which glycosidic bond can be hydrolyzed by alpha amylase? Now let's read the rules here. It only hydrolyzes 1 to 4 glycosidic bonds. It is not able to hydrolyze the last bond of a chain. It is not able to hydrolyze the bonds in a chain of 3 units attached by 1 to 6 glycosidic bond to another chain. So let's break these down. It only hydrolyzes 1 to 4 glycosidic bonds. Now, 1 to 4 glycosidic bonds is formed here between each alpha glucose molecules. These form the main chain, but not the branches. What forms this branch is actually 1 to 6 glycosidic bond. This is what formed the branch and formed this chain. Therefore, B is incorrect because this is a 1 to 6 glycosidic bond. Now let's move on to the next one. It is not able to hydrolyze the last bond of a chain. So the last bond of a chain is D, therefore D is incorrect. So B and D are both incorrect. It is not able to hydrolyze the bonds of a chain of 3 units attached by 1 to 6 glycosidic bond to another chain. So this chain, we said it's a branch attached by 1 to 6 glycosidic bond to the main chain. Therefore, here it says not able to hydrolyze the bonds in a chain of 3 units, 1, 2, 3, attached by 1 to 6 glycosidic bond. Therefore, A is incorrect And the answer is going to be C. Question number 10. Molecule X is a lipid. Which row is correct for molecule X and a triglyceride? So triglyceride is formed of glycerol attached to 3 fatty acids by an ester bond. Now, molecule X, we can clearly here see an ester bond. Therefore, this must be an alcohol and a carboxylic acid. If we were to draw these out, it would have looked something like this before condensation reaction, this molecule, and the carboxylic acid would have looked something like that. This is this molecule right here. If these undergo a condensation reaction, the OH from the alcohol, which is this part, which is the alcohol, takes one O molecule from the carboxylic acid, and then it forms an ester bond, which looks something like this. Now we know the basis of the reaction. Now let's see the suggestions that we have here. Molecule X contains 1 unsaturated fatty acid. So unsaturated fatty acids have double bonds in the main chain. Here we don't see any double bonds. Therefore, there are no unsaturated fatty acids. B. No ester bonds. This is incorrect. We've just discussed that there's an ester bond here, so this is incorrect. C. 1 fatty acid. So, as we mentioned that this molecule here is a carboxylic acid. Therefore, this is correct. D. 2 fatty acids. This is incorrect. We can only see 1. Triglyceride contains saturated fatty acids. Yes, it could contain both saturated or unsaturated fatty acids. 3 ester bonds. This is correct because each fatty acid, we said that we have 3, are joined to that glycerol molecule, each by 1 ester bond, therefore makes 3 ester bonds. Therefore, this is correct. 3 fatty acids. Yes, this is correct. And this is correct. Therefore, the only correct answer here is C. Question number 11. Bread contains a mixture of polypeptides known as gluten. 2 of the polypeptides that are found in gluten are glutenin and gliadin. Which statement describes the tertiary structure of a protein? So, let's start with the primary structure. When we talk about the primary structure, we talk about the sequence of amino acids in a polypeptide chain, which is limited to the peptide bonds. When we talk about the secondary structure, we talk about the hydrogen bonds between the nitrile group of the amino acid, which is this one, and the carboxyl group of the amino acid. And when we talk about the tertiary structure, we talk about the hydrophobic, hydrogen, ionic, and disulfide bonds formed by the R group of the amino acids this time. Be careful, not the nitrile or the carboxyl. So when we talk about the tertiary structure, we talk about the bonds formed by the R group. Now, which statement describes the tertiary structure of a protein? A. Disulfide bonds formed between glutenin and gliadin. Now, this is incorrect because glutenin is a separate polypeptide chain and gliadin is another separate polypeptide chain. When we talk about multiple polypeptide chains, we're talking about the quaternary structure, not the tertiary anymore. Therefore, A is incorrect. B. A large proportion of amino acids in gliadin are glutamine. So when we talk about amino acids, we're talking about the primary structure. Therefore, this is incorrect. C. Alpha helical sections are found in glutenin and gliadin. So when we talk about alpha helical sections, we talk about hydrogen bonds, therefore about the secondary structure. Therefore, this is also incorrect for D. Amino acids with the hydrophobic R groups are found in the inside of glutenin. So when we talk about hydrophobic R groups, we're talking about the R groups and these bonds, which are the tertiary structure. Therefore, D is the correct answer. Question number 12. A student was provided with 2 test tubes, one containing 10 cm cubed of solution P and one containing 10 cm cubed of solution Q. When these solutions were mixed together and left for 24 hours, the concentration of P decreased, but the concentration of Q remained the same. So P decreased and Q the same. The student wrote the following conclusions. Which conclusions could be supported by the information? So conclusion 1, P may break down over time. Yes, because it decreased, it might actually break down. So this is correct. 2, Q may be a biological catalyst. By biological catalyst, we mean an enzyme. And we know that enzyme, the properties of enzyme, they remain constant. They don't actually decrease in numbers. Therefore, this could be also correct. 3, P may be the substrate for Q. So Q, if we assume that it's an enzyme, P may be the substrate that actually does bind to that enzyme. Therefore, it does decrease because it's catalyzed by that enzyme. Therefore, all 3 conclusions might be correct, and the answer is A. Question number 13. Which statements are correct for the lock and key hypothesis and induced fit hypothesis of enzyme action? So, lock and key hypothesis assumes, let's draw this out, assumes that the substrate is exactly complementary to the active site. This is lock and key hypothesis. Induced fit hypothesis assumes that once the substrate, which could be a different shape, binds to the active site by some weak bonds, what happens is this active site slightly changes shape to become more complementary and allow the substrate to bind to the active site. Now let's see the suggestions that we have here. 1, the substrate is the same shape as the active site. In this case, it's only for the lock and key hypothesis, not for the induced fit. As we said, induced fit, the active site actually changes shape to become more complementary and match the substrate. Therefore, this is incorrect. For 2, the substrate is held in place in the active site by temporary bonds, which are weak bonds. This is correct. They cannot be catalyzed if they're actually not holding on. For both. 3, the enzyme and sometimes the substrate change shape slightly as the substrate molecule. Now we said what changes shape is in the induced fit hypothesis only. So this is incorrect, and the correct answer must be C. Number 14, a student investigated the rate of enzyme activity with increasing substrate concentration. So the substrate is increasing. The experiment was repeated with the addition of reversible non-competitive enzyme inhibitors. So don't be confused. It's still a non-competitive enzyme inhibitor which binds to an allosteric site, changing the shape of the active site, and it attaches permanently. And no matter how much you increase the substrate concentration, the rate of the reaction would not increase. So I hope I made this clear. Now, which graph represents the results of the 2 experiments? So the 2 experiments are without the enzyme inhibitor and with the enzyme inhibitor. So let's focus with this one. With the enzyme inhibitor, we've just mentioned that this is non-competitive. Therefore, it binds to an allosteric site, changing the shape of the active site permanently, not allowing the reaction to the rate of the enzyme activity to increase. Even if you keep increasing the substrate concentration, it still remains constant. Therefore, if we look at A, here we could see that the rate of reaction is keeps increasing as you increase substrate concentration. Therefore, this is incorrect. This would be correct if it was a competitive enzyme inhibitor because this is reversible. Now for B, we could see that it increased slightly and then was plateau. No matter how much you increase the substrate concentration, the rate did not increase. Therefore, this is correct. Now, for C, we could see that the rate of the reaction increased even more than without the enzyme inhibitor, which does not make any sense. Therefore, this is incorrect. For D, here we could see that the rate with the enzyme inhibitor was less. However, it still did reach the end point the same rate as without the inhibitor. Now, in this case, this is incorrect because, as we said, that no matter how much you increase the substrate concentration, the rate would not increase further. This would be correct if this was a competitive inhibitor. Competitive inhibitor could be reversed by increasing substrate concentration. Therefore, the only correct answer is B. Question number 15. A cuboidal epithelium cell has a length of 2 micrometers. The cell is an approximate cube shape. What is the most accurate estimate of the surface area to volume ratio of this cuboidal epithelium cell? Now, calculate the volume of the cube which is 8 micrometers, and calculate the surface area, which is 24. The simplest ratio in this case would be 3 to 1. So volume to surface area, or surface area to volume ratio. Therefore, it's going to be 3 to 1, and the answer is going to be D. Question number 16. Which statement is correct for facilitated diffusion and active transport? So let's define these terms. Facilitated diffusion is the diffusion of a substance down its concentration gradient, however, through a transport protein. Because the cell membrane is not actually permeable to that substance, therefore it does require a transport protein, but it still goes down its concentration gradient, and this is a passive and does not require energy. Regarding active transport is the movement of molecules against its concentration gradient from low concentration to high concentration through a transport protein. This requires a transport protein. However, this is an active process because it's against concentration gradient and requires ATP. Now let's see the suggestions that we have here. A. As the direction of the concentration gradient changes, so does the direction of movement of the molecules. This would only be correct in the case of facilitated diffusion because we said it's passive and it goes from high concentration to low concentration. But in case of active transport, even if the gradient changes, it still only goes in one direction. Active transport only occurs in one direction. Therefore, this is incorrect. B. Molecules always move at the same rate as simple diffusion. Now, active transport, we've said that it is an active process which requires ATP. Therefore, it makes more sense that active transport actually occurs faster than a passive mechanism such as diffusion or facilitated diffusion. Therefore, this is incorrect. C. Specific molecules are transported across a membrane. So this is correct because we've just mentioned that both of them require transport proteins. These transport proteins are highly specific for specific molecules. Therefore, this is correct. D. The molecule ATP is required to move specific molecules quickly through proteins in the membrane. As we said, this is only correct for active transport. Therefore, this is incorrect, and the answer is going to be C. Question number 17. Equal-sized potato pieces were placed into a test tube and covered with sucrose solution. The test tube was left for 30 minutes. All other variables were standardized. After 30 minutes, the potato piece has not changed in size. What can be concluded from this result? So, the potato piece has not changed in size. This is a clear indication that both the sucrose solution and the potato have the same water potential. Be careful, when we talk about osmosis, or movement of water, we talk about water potential. Both of these have the same water potential, therefore there has been no net movement of water. Now let's see the suggestions that we have here. A. The concentration of sucrose is the same in the potato. Now, we said when we talk about osmosis, this is a common error between students. We never talk about the concentration of sucrose. We always talk about the water potential. Therefore, both of these are incorrect. Now, for C, the water potential is the same in the potato and in the sucrose solution, and there is no more movement of water into or out of the potato. This is totally incorrect. The reason for this is there is still movement. However, they happen at the same rate. There's no net movement. Therefore, there's not more water maybe that goes out than in. They both move at the same rate equally. So this is incorrect. But movement does not stop, because as we all know that particles are always moving. D. The water potential is the same in the potato. This is correct. And in sucrose solution, and there's no net movement of water into or out of the potato. So there's no net movement. This is the correct expression. Therefore, the answer is D. Question number 18. How many of these processes produce genetically identical cells? Now, growth of plant roots. Yes, plant roots occurs by mitosis to regenerate. Therefore, this is correct. Now, the next one, replacement of dead human skin cells. Also by mitosis. Therefore, it does provide genetically identical cells. This is correct. Now, repair of damaged muscle tissue. So, medically, it depends. It depends on the extent of damage. If there's too much damage, then it repairs by something called fibrous tissue, but no need to know a lot of details about this. So, medically, this is not always the case. However, in the sake of the syllabus, this is correct. Repair of damaged muscle tissue is by producing genetically identical muscle cells. Now, asexual reproduction of coral. So asexual reproduction does produce genetically identical cells, so this is correct. Therefore, the answer is D. Question number 19. Which row shows the correct number of each component of a single chromosome at the end of prophase of mitosis? So, we know that in the mitotic cycle, there is something called the S phase, G2, G1. This is the interphase. And there is mitosis, which consists of prophase, metaphase, anaphase, and telophase. Now, we know that in S phase, there's the replication of DNA. Therefore, at the start of mitosis during prophase, there's going to be 2 copies of everything. Now, let's see suggestions that we have here and to elaborate more on what I mean. Now, centromeres. Centromeres are in the center between each chromosome molecule, which has the kinetochore, which the spindle attaches to during anaphase and separates them apart. So, centromere is present, only one, in a single chromosome. Therefore, these are correct and these are incorrect. Now, chromatids. As we said, that during DNA replication, the single chromosome unit has replicated into 2. Therefore, each one of these is called a chromatid. Therefore, after the S phase and at the start of prophase, we would have 2 chromatids. Therefore, this is correct. And all these are incorrect. Now, polynucleotide strand. Each chromatid has 2 polynucleotide strands. So that's what it would look like if we would draw them out. So each chromatid has 2 polynucleotide strands. Therefore, a single chromosome would have 4 polynucleotide strands. Therefore, this is correct. And telomeres. We know because we have 2 chromatids, each chromatid has 2 telomeres at its end. Therefore, we have 4 telomeres and the answer is B. Question number 20. Which processes occur during interphase? So during interphase, we have the G1 phase, the S phase, which DNA replication occurs, and the G2 phase, which is the growth of the cell. Now, what occurs during interphase? DNA replication, we said, is during the S phase. So this is correct. 2. Microtubule organization. So microtubule organization actually occurs during mitosis by the centrioles inside centrosomes. So these are the centrioles inside the larger ones, which are called centrosomes. Let's assume that this is a giant cell. These centrioles produce the microtubules. Let's assume that this is the spindle fiber. So the centrioles organizes microtubules into a spindle, which attaches to the kinetochore of a chromosome, pulling them apart during anaphase. Therefore, this is incorrect. Firstly, synthesis of a ribosomes. So we know that during interphase, we have the G1 and the G2 phase. The G2 phase, the cell continues to grow. How does it grow? It does grow by ribosomal RNA synthesis, by the synthesis of proteins, etc., etc. Therefore, part of this growth obviously involves the synthesis of ribosomes. Therefore, this is correct, and therefore the answer is going to be C. Question number 21. Embryonic stem cells are able to replicate continuously. What happens to the telomeres during repeated mitotic cell cycles of embryonic stem cells? So let's define these terms. So embryonic stem cell is a pluripotent stem cell, meaning it could form any type of cell. No matter what it is, it could form any type of cell. Now, stem cells, we know that they have an enzyme called telomerase, which synthesizes telomeres. This feature allows stem cells to replicate or divide, continuously or unlimited number of times. Therefore, let's see the suggestions that we have here. A. Their length increase. This is incorrect because telomere length remains constant in stem cells because of the telomerase enzyme. They don't actually increase more than it should. Their lengths decrease. This would happen in any typical cell, however, not stem cells. Therefore, this is incorrect. C. They're completely lost. Also, this is incorrect. D. They stay the same length. This is correct, and the answer is going to be D. Question number 22. Which nitrogenous bases are pyrimidines? So, purines are adenine and guanine. Pyrimidines are thymine, cytosine, and uracil. Therefore, the correct answer is going to be C. And by the way, purines are made of 2 rings, while pyrimidines are the smaller ones made of a single ring. Question number 23. DNA replication in bacteria was investigated. Bacteria were grown in a medium with only heavy nitrogen, N15, until all the bacterial DNA was heavy. These bacteria were moved from heavy nitrogen medium and cultured in a medium with only light nitrogen, N14. This formed the first generation. The bacteria continued to reproduce, and a sample of bacteria was collected from the second generation and the DNA analyzed. Hybrid DNA contains heavy and light DNA. So, a hybrid DNA contains 1 chain made of N14 and 1 chain made of N15, which row is correct for the second generation. Now, this question or these types of questions are always addressed in the next method that I'm going to show you right now. First of all, we're starting with N15, N15. And what we're doing to actually receive the first generation is that we're introducing N14. Now, what's going to happen is we're going to have 2 hybrid strands made of N15, N14, and N15, N14. Now, this is our first generation. Now, let's move on to the second generation. We're introducing more N14. Now, what's going to happen is that we're going to have 4 different DNA. So we're going to have N15, N14, N14. N14, N15, N14, and lastly N14 and N14. Now, let's see the suggestions that we have here. First of all, percentage of heavy DNA strands. So, the heavy strands are N15. So, we have 1 and 2. 2 out of 8. Therefore, we're going to end up with 25%. Therefore, 25 is the correct suggestion. And percentage of hybrid DNA molecules. We've discussed earlier that hybrid DNA molecules contains both N15 and N14. Here, we have 50%. Because we have 1 here, N15 to N14, and N15 and N14, making it 50% of the total. Therefore, the answer is going to be B. Question number 24. The sequence shows the series of bases at the start of a gene. After transcription, the mRNA was translated via tRNA into a sequence of amino acids. When this part of the polypeptide was analyzed, it was found to contain the amino acids in the table. What is the sequence of amino acids in this part of the polypeptide? Now, in this question, we mostly just have to use common sense. Now, here we have the amino acid and the number of amino acids present. And we know that a single amino acid is made of 3 bases. Therefore, here we have glycine, which are 3 glycines present. Therefore, it's most likely going to be CCA, CCA, and CCA. And here for alanine, we have 2 present. Therefore, most likely, this is going to be the alanine. Therefore, we could clearly see that B is going to be the correct answer. Question number 25. The table shows the mode of action of two antibacterial drugs that can affect the synthesis of proteins. If bacteria are treated with the drugs rifampicin and streptomycin, what will be the immediate effects? So the short-term effects. Now, let's see the suggestions that we have here. 1. Transcription will stop, but faulty proteins may continue to be synthesized. So let's go back to the mode of action of these drugs. So rifampicin binds to RNA polymerase. Therefore, binding to RNA polymerase stops the formation of messenger RNA. Therefore, this stops translation of proteins. Now, streptomycin causes errors in translation. So transcription will stop, but faulty proteins may continue to be synthesized. Yes, because yes, we did stop mRNA formation. However, there are still mRNA molecules in the cytoplasm. Therefore, they can still be translated, but due to the streptomycin, there will be errors in that translation. Therefore, this is correct. And this is a short term because we're assuming that there are still mRNA in the cytoplasm. Now, 2. If translation has started, proteins may be faulty. Yes, this is correct because we're going back to the first point, assuming that there's still mRNA in the cytoplasm. Therefore, this is correct. Now, 3. Translation will be inhibited. Now, this is not correct because this is in the long term, not in the short term. Translation will be inhibited when there are no more mRNA molecules in the cytoplasm on the long term. Therefore, only 1 and 2 are correct. Therefore, the answer is going to be B. Question number 26. The electron micrograph shows a cross-section of a leaf vein and some neighboring cells. Which structure contributes to mass flow by the use of proton pumps? First of all, you have to notice that this is a transverse section through a leaf. And you should be able to locate the positions of the phloem and the xylem in a leaf. So usually, the phloem is located at the bottom, and the xylem is located upwards. Now, which structure contributes to mass flow by the use of proton pumps? Now, first of all, you must notice that sucrose movement through the phloem is actually an active process which requires proton pumps in the companion cells to pump hydrogen ions outside because it is an active process which requires ATP. Therefore, here we're looking for the phloem. And because we've mentioned that phloem is at the bottom middle, therefore the answer is going to be C. Number 27. The diagram shows some tissue types in plant organs. Which row identifies tissue types? So, first of all, this is a root, a transverse section through a root. And you must know that in a root, the xylem is the cross sign and the phloem is surrounding it. Therefore, 1 is definitely the phloem. Now, for this photo, this is a transverse section through a stem. And you should know that the outside in a stem is always the phloem. And the inner part, which is this part, is the xylem. Therefore, 2 is also the phloem. Now, 4 and 3. First of all, you must know that this is a transverse section through a leaf. And as we mentioned in the other question, that the upper part is going to be the xylem and the lower part is the phloem. Therefore, 3 is the phloem and 4 is the xylem, and the answer is going to be A. Question number 28. Which description of xylem vessel elements is correct? A. Cells join to form a tube. Pits at intervals. So, forming a tube is correct because xylem vessels is a hollow tube. Pits at intervals, this is correct. It has been known that these pits are mainly to stop air bubbles. Now, sieve plates between cells. This is incorrect because we've just mentioned it's a hollow tube and surrounded by endodermis in roots. Now, this statement is incorrect. Why? Because in the last question we've discussed that in the roots, the xylem looks something like a cross. So this is the xylem, and what's surrounding it is the phloem. Therefore, what's surrounding the xylem in the phloem is, in the roots is actually the phloem, not the endodermis. The endodermis is what covers both the phloem and the xylem. Therefore, this is incorrect. For B, contains cells joined end to end, containing cytoplasm, cell walls. Now, first of all, Containing cytoplasm is definitely incorrect because we've mentioned that xylem is a hollow organ. Therefore, it's dead. So it does not contain cytoplasm. Contrasting phloem. Phloem actually does contain cytoplasm. So this is incorrect. C contains elongated cells with end walls broken down located in vascular bundles in a stem and leaves. Now, we've just mentioned that it's a hollow tube, so this is correct. Located in vascular bundles is also correct. For D, dead elongated cells, yes. Lignified cell walls with pits at intervals. This is also correct. Associated with companion cells. Now, companion cells is incorrect because companion cells are associated with the phloem to facilitate the transport of sucrose into the phloem. Therefore, this is incorrect, and the answer is going to be C. Question number 29. Which properties of lignin are important for the function of xylem vessels in the stem of a tall plant, such as a tree? So here the question is asking about supporting functions of lignin. Now 1. It is inflexible, so does not bend easily. Yes, this is correct because it's one of the supporting functions. For 2. It is not permeable to water. Also, this constitutes to the support, so this is correct, and it is actually impermeable to water. 3. It is strong to resist collapse under pressure. This is also a supporting function. Now 4. It has weaker adhesion to water molecules than cellulose. Now this is incorrect. Why? Because lignin actually has a higher affinity to OH groups in water than cellulose. Therefore, it has a stronger adhesion. So 4 is incorrect, 1, 2, and 3 is correct. So the answer is going to be A. Question number 30. Which row is correct for the sucrose concentrations and the water potentials in a source and a sink in a potato plant at 12 p.m. So first of all, you must notice that at 12 p.m. photosynthesis is actually taking place. Now, here we have the sucrose concentrations and water potential. And because photosynthesis is taking place at 12 p.m., the source in this case is going to be the leaves, and the sink is going to be the potato plant. Now, in this question, they're asking about the sucrose concentration. So, due to the photosynthesis, the sucrose concentration at the source is definitely going to be higher. Now, for the water potential, this is where the confusion comes in. The water potential will be higher. You might ask why. That actually there is a higher concentration of solute at the source. So how could the water potential be possibly higher? Now this is for 2 reasons. The first of all, when the sucrose concentration increases in cells in the leaves, what happens is that water from neighboring areas goes inside these cells down water potential gradient. Therefore, what happens is even if the sucrose concentration in these cells is increasing, water is also entering down water potential gradient. Therefore, the water potential inside cells actually increases, and turgor pressure obviously increases. And the second reason is that at 12 p.m. the stomata is actually open, therefore allowing evaporation. Therefore, the water potential must be higher at the source. Therefore, the answer is going to be A. Question number 31. The diagram shows one possible way in which sucrose may be loaded into a sieve tube element. Which steps require ATP? So, first of all, the steps in the companion cell is what requires ATP because, as we all know, sucrose movement through the phloem is an active process requiring ATP. So first of all, what happens is we have a companion cell and here we have the phloem tube element. What happens is that companion cell pumps hydrogen ions to its cell wall via proton pumps. What then happens is the hydrogen and a sucrose molecule joins together and goes back into that companion cell through a co-transporter protein via carrier protein, making hydrogen ions go down concentration gradient and sucrose molecules go against concentration gradient. Therefore, what happens is that the sucrose molecules are now inside the companion cells and ready to diffuse into the sieve tube elements. Therefore, which steps require ATP? we've mentioned protons move out of the companion cells by proton pumps, which requires ATP, so the answer is going to be 1 only. Question number 32. Which statements about arteries are correct? 1. Artery walls can resist high pressure. This is correct due to the elastic fiber content of its walls. 2. Arteries pump blood out of the heart. Now, this is incorrect. It's the heart that pumps blood out of it. 3. Blood in arteries has the same flow rate as in veins. This is incorrect because as we are leaving the heart, the blood vessels with the fastest flow rate is the arteries, then the capillaries, then the slowest being the veins. Therefore, this is incorrect. 4. The pulse in arteries is the result of a surge in blood that causes expansion of the artery wall. This is correct. And 5. There are semilunar valves at the junction of arteries with the heart. This is correct because there are valves between the ventricles and the arteries, stopping the backflow of blood from the arteries back into the ventricles. Therefore, this is correct, and the answer is going to be C. Question number 33. What happens during ventricular systole in a mammalian heart? So ventricular systole is the contraction of the ventricle, and during that contraction, the pressure in the ventricle increases, pushing the semilunar valves open, and the volume in the ventricles decreases as a result of this. Now, 1. The atrioventricular node transmits an electrical signal to the apex of the heart. Now, this is incorrect. The atrioventricular node actually slows down the impulse, so the atrium and the ventricles do not contract at the same time. What actually sends the signal to the apex of the heart is the bundles of His and the Purkinje fibers. So this is incorrect. For 2, the pressure in the ventricles drops below the pressure in the atria. Now, this is not possible. In order for the ventricles to push the semilunar valves open and actually push blood into the atrium, it requires the pressure to be higher. Therefore, this is incorrect. 3, the atrioventricular valves close and the semilunar valves open. This is correct because atrioventricular valves closes due to the backflow, shutting the atrioventricular valves. And the semilunar valves open due to the increase in pressure. Therefore, the answer is going to be D. Question number 34. A shortage of protein in the diet of children can lead to a disease that causes excess tissue fluid to build up in the abdomen. What explains why a buildup of excess tissue fluid can occur in this disease? Now, first of all, you have to know that the composition of plasma is mainly protein. And by having a shortage of protein, this means that the water potential in the plasma increases, therefore causing extra tissue fluid. Therefore, tissue fluid accumulates, causing buildup in the abdomen. Now, let's start with A. A shortage of plasma proteins decreases water potential. This is incorrect. It increases the water potential. B. A shortage of plasma proteins increases blood water potential. Less tissue fluid returns to the blood. This is correct because tissue fluid is stuck at the organs, such as the abdomen. Therefore, less of it returns to the blood. Therefore, edema happens. C. A shortage of protein in the diet causes weak cardiac muscle, reducing hydrostatic pressure. Now, weak cardiac muscle has something to do with the cardiac output. Cardiac output is to do with the force of pushing blood out and the pressure. Therefore, it has nothing to do with the hydrostatic pressure. By talking about the hydrostatic pressure, we must mention the solute concentration as the key factor. Therefore, this is incorrect. A shortage of protein in the tissue fluid decreases its water potential. We just mentioned this is incorrect. Therefore, the answer is going to be B. Question number 35. Red blood cells mainly contain a molecule known as 2,3-bisphosphoglycerate. When 2,3-bisphosphoglycerate binds to hemoglobin, a higher partial pressure of oxygen is needed to bring about 50% saturation of a hemoglobin with oxygen. This clearly indicates that 2,3-bisphosphoglycerate decreases the affinity of hemoglobin to oxygen. Therefore, what happens is that the curve shifts to the right. Why? Because now a higher partial pressure is needed to achieve the same saturation. Now, which statements about the effect of 2,3-bisphosphoglycerate are correct? 1. It causes the oxygen dissociation curve to shift to the right. This is correct because it decreases the affinity of a hemoglobin to oxygen. 2. The binding of 2,3-bisphosphoglycerate to hemoglobin reduces the Bohr effect. Now, first of all, when we talk about Bohr effect, is a decrease of affinity of hemoglobin to oxygen due to the presence of carbon dioxide. So first of all, this is incorrect because when we talk about Bohr effect, we talk about the carbon dioxide concentration. Second of all, if we assumed that the affinity of hemoglobin to oxygen actually decreased, therefore it would actually increase the Bohr effect, not reduce it. Therefore, it's wrong in both ways. 3. The binding of 2,3-bisphosphoglycerate to hemoglobin lowers the affinity. Obviously, this is correct. 4. When 2,3-bisphosphoglycerate is not present, oxyhemoglobin is less likely to unload oxygen. First of all, when we're talking about this molecule here, we're talking about the affinity of hemoglobin to oxygen. We're not even mentioning oxyhemoglobin. Second of all, we're mentioning the loading of oxygen, the affinity of hemoglobin to oxygen in the loading, not the unloading. Therefore, 4 is incorrect, and the answer is going to be question number 36. Carbon dioxide is continually produced by respiring cells. It diffuses into red blood cells and dissociates into hydrogen ions and hydrogen carbonate ions. Hydrogen carbonate ions then diffuses outside the cell in exchange of chloride ions in the chloride shift. What is the importance of the chloride shift? First of all, chloride shift is the movement of chloride ions into red blood cells from blood plasma to balance the movement of hydrogen carbonate ions into the plasma from red blood cells. First of all, you must know that inside a red blood cell there is an enzyme called carbonic anhydrase. This enzyme basically forms carbonic acid. What happens is the reaction combining carbon dioxide with water forming carbonic acid. Then what happens is that carbonic acid dissociates into hydrogen ions and hydrogen carbonate ions, which are negative. Now, hydrogen ions are actually impermeable. They cannot leave the red blood cell. What happens is that the hydrogen carbonate ions are able to leave and enter the blood plasma. But what happens is we mentioned that hydrogen carbonate ions are negatively charged. Therefore, what would happen if we just left the hydrogen carbonate ions to leave the red blood cell? What would happen is the positive charges would accumulate. The red blood cell would have a net positive charge. So, in order to balance this phenomenon, what happens is that the chloride ions, which are negatively charged, are actually permeable and can enter the red blood cell to balance these charges. Now, let's see the suggestions that we have here. A. It helps to maintain a neutral pH. This is incorrect. We're talking about the positive and negative charges. B. It maintains a balance of positive and negative charge between the cell and the plasma. This is correct. C. It causes the oxygen dissociation curve to shift to the left, releasing more oxygen. This is incorrect. It prevents carbon dioxide combining with hemoglobin, allowing hemoglobin to combine with oxygen. This is incorrect. Carbon dioxide combines with the globin part of the hemoglobin, and it has nothing to do with the chloride shift. Therefore, the answer is going to be B. Question number 37. The photomicrograph shows a section through a bronchus. Which annotated label is correct? Now, let's start with correctly labeling the photo. First of all, D. Squamous epithelium shows for efficient gas exchange. Squamous is incorrect. These cells are called pseudostratified columnar ciliated epithelium. They are columnar, not squamous. Therefore, this label is incorrect. For C, it's assuming that this is cartilage. Cartilage keeps the airways open. This is incorrect. The reason why is if we zoom in deeper, this is actually a mucus gland, which has cuboidal cells, as you can see here. Now let's go to B. Elastic tissue stretches when breathing in. This is also incorrect because you can clearly see that this is hyaline cartilage. This is not elastic tissue. Now lastly, smooth muscle changes the diameter of the airways. Yes, underneath that layer of pseudostratified columnar ciliated epithelium, there is a layer of smooth muscle. Therefore, A is correct. Question number 38. Penicillin is an antibiotic used to treat infections caused by bacteria. Which events explains how bacteria become resistant to penicillin? 1. The number of bacteria with the allele for the resistance to penicillin increases. This is correct. There are the resistant genes in the bacteria which give it resistance to penicillin. 2. Resistance to penicillin is the result of a mutation. Yes. Any resistance is a cause of gene mutations which occur naturally in the environment. 3. Bacteria with the allele for their resistance to penicillin pass the allele to their daughter cells. This is definitely correct. 4. The mutation for resistance to penicillin is always caused by the presence of penicillin. This is incorrect. The mutations happens normally and they happen due to a change in the base sequence of the DNA. Therefore, this is incorrect and the answer must be B. Question number 39. Which statements explain why some vaccines can be taken by mouth, but tuberculosis vaccine has to be injected? 1. Macrophages presents antigen in vaccines to stimulate an immune response. This is correct. However, it does not explain why some vaccines can be taken by mouth, but tuberculosis has to be injected. Therefore, this is incorrect in the context of the question. 2. The tuberculosis antigens necessary to produce an immune response are proteins, which would be digested in the stomach and the small intestine. This is correct. If the proteins are digested by enzymes in the gut, what happens is that antigen presentation cannot take place anymore. Therefore, a primary immune response cannot be generated. Therefore, there are no memory cells. Therefore, the vaccine has no use. Therefore, this is correct. 3. There are no B lymphocytes and T lymphocytes in the stomach. This is definitely incorrect. The gut is filled with intraepithelial lymphocytes to fight off any microorganisms found inside our meals. Therefore, this 3 is definitely incorrect and the answer is going to be D. Question number 40. A student wrote the following statements about the production of monoclonal antibodies. Which statements is not correct? Now, the process of monoclonal antibodies is that non-self antigens is injected into a mammal. This is correct. For example, a rabbit. And then what happens is that rabbit generates a primary immune response. And plasma cells form antibodies. Now, B. Plasma cells secretes a specific antibody in response to a non-self antigen. This is correct. C. Selected hybridoma cells divide by mitosis to form a clone. So hybridoma cells are a fusion of cancer cells and plasma cells. And then these fused cells divide by mitosis to form a clone of hybridoma cells. So this is correct. D. T lymphocytes are fused with cancer cells. Now this is incorrect. We've just mentioned that plasma cells are fused with cancer cells to form hybridoma. Therefore, this is the incorrect answer and the answer is going to be D.